Hello, everybody, and welcome to our first official lecture of Bio 50B, Spring 2021. I wanted to walk you through a little bit of our Canvas page a little bit before I get started on Chapter 11. So we finished up week 1. There is a lecture assignment that will be available to you until the 28th. So that's just for people in case we get a chance to add somebody this next week. This is, again, your introduction to MyLab and Mastering. If you are having trouble getting MyLab and Mastering up running, please troubleshoot first by going to the help and support link. You've got welcome to mastering, just some basic like system requirements kind of stuff that you might be missing out on. And some more helpful stuff at this link here. This is available to everybody. You don't actually have to have it. Once you hit open MyLab and Mastering, that's when you're going to need to log in or register or purchase your access. So if you're having trouble with MyLab and Mastering, you should be able to just click this link here and get here and go to help and support. Okay. This week, week 2, in our to-do list, we have this week's reading, chapter 11. We've got these lectures that you are viewing right now. This is lecture 1 of 2 for covering chapter 11. The homework for this week due on Friday is right here. This is what we're going to be doing on Thursday. Lots of details in there. And this is all just additional fun for you stuff to do to help you to learn chapter 11, okay, to study chapter 11. So this is basically everything that you need to do this week, all right, is in week 2. If you do everything, including all of the additional resources, I can pretty much guarantee that you will be fine on any exams on this material. If you do all the reading, do all the homework, watch the lecture videos, and look through all of these additional resources, like actually look at them and do them, then you will be fine, okay? So that's why that's there. So check it out. All right, chapter 11, we're picking up where we left off in part B, or part A, 50A, when we talked about the muscular system and some of the special senses. So we're actually kind of backtracking a little bit to talk about the general nervous system and how it works. So this time we're going to be talking about the nervous system as a organ system. And in this chapter, we're going to be looking at the fundamentals of nervous tissue, what it does, what it looks like, and how it works. So let's get started. So your nervous system is by weight the smallest organ system of the human body. It depends on electrical activity, right? That electrical activity comes from ions, right? Cations and anions. These are negatively or positively charged atoms because they have either taken an extra electron or given away an electron. That charge, that plus or minus charge, that is what is the electricity. That's what causes the electricity that your nervous system is able to conduct to send signals by. And we will talk all about that in the part 2 of this lecture. Your nervous system works hand in hand with your endocrine system to send signals to all of your body to control your entire body. Okay, so to do all body functions, it's a combination between either your nervous system or your endocrine system. They're both communication systems in your body. One works by electrical activity, and the other works by chemical or hormone activity, right? Okay, so the basics about your nervous system, the basics of how it works, right? Your nervous system functions by taking sensory information, processing it, and then responding to it. And we have special names for like these different parts of it, okay? That sensory input from a sensory organ, whether that be touch receptors in your skin or the photoreceptors in your eyes, the hair cells in your inner ear, the cells of any other of your senses, your smell and taste senses, or sensors within your body having to do with homeostasis, right? So sensors that detect your body temperature, sensors that detect your blood pressure, sensors that detect whether your stomach is empty or full. All of these things are sensory, and all of that information gets relayed to the central nervous system by afferent nerves, okay? By the afferent portion of your peripheral nervous system. So the sensory input is also known as afferent. Integration of that information happens in the central nervous system, also known as the CNS, right? I think I got an extra line in there somewhere. Oh, no, actually, oh, yeah, okay. Once that information is integrated and your brain makes a decision about what it's going to do about that sensory input, about that stimulus that it just received, the information that goes from the central nervous system back into your peripheral nervous system to control your muscles or control endocrine glands or to control visceral organs inside of your body, that pathway is known as efferent. One way you can try to remember this is that afferent, A comes before E in the alphabet, okay? So the afferent pathway happens first. The afferent pathway is the sensory pathway. The efferent pathway goes to effectors, which can include muscles, glands, or organs, okay? And that is the pathway that controls your body's response to the signal or to the stimulus, okay? You've got two main divisions of your nervous system. You probably already know this. The central nervous system is made up of your brain and your spinal cord. Okay, so those are in gray or purple here. And your peripheral nervous system, or the PNS versus the CNS of your central nervous system, is all of the nerves that branch off of your spinal cord to go and feed all of your muscles, glands, and organs, and that come from all of your sensory receptors throughout your body, okay? So your peripheral nervous system both provides the sensory info to the central nervous system, as well as carrying the signal from the central nervous system back to the effectors that control your response to that sensory info, okay? So it carries motor commands to peripheral tissues, and by peripheral tissues, that can be, again, muscles, glands, or organs. And it is in yellow, goldish yellow here, okay? So central nervous system, brain and spinal cord, peripheral nervous system, all the rest of your nerves in your body, okay? Both sending information to the CNS and from the CNS to your body. Okay. Your peripheral nervous system, as we just discussed, has two main branches, the afferent and the efferent. Okay, so both of those are technically the peripheral nervous system, right? Both this part here and this part here are outside of the central nervous system. Okay, so both the afferent and the efferent are part of the peripheral nervous system. The afferent, again, is responsible for sending sensory information from your sensory receptors to the central nervous system, to the brain or the spinal cord. And the efferent division is responsible for sending the information from the central nervous system, from the brain, back to effectors, which can include muscles, glands, or organs, to respond to that sensory information from the afferent division, okay? Your efferent division has, again, two subdivisions, okay? There is your somatic nervous system, which would be like your organs and glands, and your somatic nervous system, which would be your skeletal muscle, and your autonomic nervous system, which would be your organs and glands, okay? So the somatic nervous system is voluntary, right? you can control, most of the time, you can control the muscles in your body to respond to a stimulus, right? You see, you're about to cross the street and you see a car coming and you wait. That's your brain telling the muscles of your legs not to walk, right? Based on that sensory input that you received from your eyes. The autonomic nervous system, still part of the efferent system, is not voluntary, right? So this is going to be glands and organs. Okay, so the autonomic nervous system, it's automatic, if you will. Okay, so don't get those two words mixed up, but auto as in like automatic, but autonomic nervous system is the part that controls the involuntary functions to maintain homeostasis within your body. So that's what's going to be sending information from the brain to tell your heart to beat, to tell your lungs to breathe, to tell your glands to produce the hormones, to tell your organs to secrete what they need to secrete and do what they need to do to maintain your life, right? The autonomic nervous system also has two subdivisions, which we're not going to talk about right at this minute. But I want to mention them, of course. The sympathetic and the parasympathetic nervous system. Okay, these guys are both involuntary, but they have to do with different ways of being alive, right? The sympathetic nervous system is that which is responsible for maintaining survival in moments of stress. Okay, so the sympathetic nervous system is going to basically it's going to basically shut down your digestion and other non-absolutely essential body systems when you are in danger. And your parasympathetic nervous system is going to be the one that actually activates those things when you are at rest. Okay, so they work hand in hand and they're kind of complementary to each other as well as they're oppositional to each other. So when one is active, the other one needs to be tampered down. And when the other one is active, the other one needs to be tampered down. Okay, so they kind of work hand in hand that way. So those are both involuntary, part of the autonomic nervous system. You also have the somatic nervous system, which is voluntary. Those are both part of the efferent division of your peripheral nervous system, okay? The efferent as in sending signals out. The afferent as in receiving sensory information, which can be from your body, like the stuff that you are aware of, right? So your senses of touch and smell and taste and hearing, as well as the stuff that you're not really aware of, kind of conscious level. So this visceral sensory fibers would be your heart rate, your blood pressure, whether you're hungry or not, the things that you don't have control over. Okay. All right. So this part of the peripheral nervous system can be anywhere in your body that is maintaining homeostasis and can be changed by a stimulus. That information goes to the central nervous system, and then the signal from the central nervous system returns to the peripheral nervous system through the efferent division to either control voluntarily your muscles, like removing your hand from a hot stove, or something like a gland or an organ in the case of returning blood pressure, for example, to homeostasis, okay, to baseline, to its stable place. So here is that kind of broken down for you in case you wanted to go back to this. This is on page 392 of your textbook. If you're using the 11th edition, it shouldn't be too far away if you're not. But this is, I think, a nice visual representation of these things. So the central nervous system is just the brain and the spinal cord. It's going to integrate and integrate information, sensory information, as well as control your body. And then the peripheral nervous system has the afferent and efferent subdivisions. The efferent subdivision is further divided into the somatic and autonomic nervous systems. And the autonomic nervous system is further divided into the sympathetic and parasympathetic nervous system, okay? All right. Let's talk about nervous tissue or neural tissue. Okay, let's talk about neurons and the helper cells, the support cells, the whole system that keeps them alive, keeps them going. Pardon me. So of course, neurons are the classic cell that you imagine when you think of nervous tissue, right? And when we talked about histology back in part A, we might have only ever actually seen a neuron as an example of nervous tissue. And it's got, you know, the classic star-shaped body and the long tail with some other stuff going on there, right? You got your classic neuron. So of course, not all neurons look like this, but generally speaking, all neurons, no matter what they look like, are also known as nerve cells. They are excitable, which means that they can be stimulated to send a signal, and they are capable of transmitting electrical signals. And hang on, this is going to drive me crazy if I don't fix this right now. Okay. And we're back. All right. So those are your neurons, right? They're the ones that actually send the electrical signals. But there is a lot more to neural tissue than just the neurons. There are a whole host of support cells that help neurons to do their job and keep them safe and protect them from pathogens and heal them from injury and stuff like that in some cases. These are called neuroglia. Okay, so neuroglia include all of these different types of support cells. Neuroglia can be roughly translated into nerve glue. So the neuroglia are responsible for holding all of your nervous tissue together, like literally, physically holding it together. They do this by surrounding, as in being all around, but also touching a bunch of neurons. So they can be reaching out and holding on to a bunch of different neurons. They can be actually physically wrapped around neurons, different parts of neurons, and then they actually physically support and hold neurons in place. So while this right here is a neuron, here's its nucleus, you also have all of these other cells. Look at this guy, like wrapped around the axon of this neuron. This guy has himself wrapped around a couple different neurons over here. And then this cute little guy over here is also touching multiple neurons with all of his little processes, his little extensions, right? So let's talk about all these different types of cells and what they do, whether they help to maintain the internal environment of the neurons, whether they help to maintain the external environment of the neurons, that extracellular fluid, and whether they actually act as immune cells and phagocytize dangerous pathogens that might be trying to attack your neurons, right, or other nervous tissue. Okay, so you've got 4 main types of neuroglia in your central nervous system. Okay, this is just the central nervous system now. We've got astrocytes, microglial cells, ependymal cells, and oligodendrocytes. All very fun words to say. Try it. Let's go through them one by one. Astrocytes, which you might remember as star cells, are these guys right here. So you've got the cell body with his nucleus right here, and all of these arms, all these processes reaching out and touching multiple neurons. This looks like the axon of one neuron. This could be the axon of another, and over here maybe the dendrite of a third. So these guys are responsible for a few things. They control the interstitial environment of the cells. So that's literally the extracellular fluid that the cells are floating in. It controls whether that fluid has the right concentration of whatever for the neurons to be able to behave properly. They maintain the blood-brain barrier, right? So while they can be attached to neurons, oh, I was mistaken here. So yeah, so if they're attached to neurons or touching neurons, at the same time, they could be touching blood vessels. So these purple ones are actually capillaries. And so they maintain the blood-brain barrier by taking things from the blood and transmitting them to the neurons as necessary. So they are like the gatekeepers between your circulatory system and your cerebrospinal fluid, your extrastitial fluid of your nervous system, as well as the intracellular fluid, the cytoplasm inside of the neurons themselves. Which is extremely important because there's a lot of pathogens that they travel freely through the blood, but if they get to the brain, then you're in really, really big trouble. The blood-brain barrier prevents a lot of that from happening. They also create the physical three-dimensional framework of the central nervous system. So when you look at brain or spinal cord tissue, a lot of that, or how the neurons are actually physically arranged in space, is determined by how the astrocytes are holding them in position. They perform damage, or they perform damage. They don't perform damage. They perform repair on damaged neurons. And this is only in some cases, right? So neurons damaged past a certain point cannot be repaired, but very, very minor damage may be repaired by astrocytes. And they also guide neuronal development, especially like early on, right? So your neurons, they develop once, and then that's really kind of it. So in the beginning, when you're developing your brain, these guys are going to actually help to physically guide the axon of one neuron to the body of another, so that a signal could be transferred between the two. So it actually like helps to form the pathways, the synaptic pathways in your brain, which is pretty awesome. Microglia kind of look like a weird starfish, almost like a brittle star, if you've ever seen a brittle star. They're very tiny. They're the tiniest of these neuroglia. They also are going to be reaching out and touching multiple neurons with their processes. They are going to also be able to, however, move around. So these guys actually can physically move around and rove around the central nervous system between the neurons, and they are able to detect whether or not a neuron is in danger or in trouble, having a hard time, damaged, any of these things, and it is able to help that neuron by maybe doing some repair. It monitors the neuron's health. It may call immune cells in, or other repair cells like astrocytes in to do repair work. And they also are going to detect pathogens in and around the neurons. And when they do, they can actually transform into phagocytes, which, if you recall from part A, are cells that are able to engulf and eat other cells, namely pathogenic cells, right? So they can actually engulf bacteria or viruses that they encounter in the central nervous system. So microglia, super important. Ependymal cells. So in your brain, you know how you've got the convoluted folds of your brain. So that's on the outside, and then also on the inside of your brain, there are spaces in there that are separate from the brain itself. They're outside of the brain, right? But they're still very, very important, and they're still wrapped in like the visceral membrane that surrounds the entire brain. So these are kind of like little hollow places in there. We call them ventricles, and they are full of the cerebrospinal fluid. The ventricles and the cerebrospinal fluid inside of them need to remain intact, right? Because they are performing important immune functions for the brain, as well as physical support and protection, cushioning kind of stuff for the brain. So it's really important that that fluid stays in there and that it stays just the right volume in there, right? The ependymal cells are actually a type of epithelium that lines the ventricles of the brain. And these guys can be squamous shaped, right? They could be squamous cells, they could be cuboidal, or they can be columnar. And regardless of what they are, the shape that they have, they're usually ciliated, and they are going to be held together by why am I keeping this? Tight junctions that prevent the cerebrospinal fluid from leaking out in between them, right? We learned about tight junctions when we talked about histology in part A. They hold cells together, epithelial tissue, very tightly, and they're usually ciliated. So the cilia of these ependymal cells are going to beat, right? Because that's what cilia do. They're supposed to be waving, and they're usually moving fluid around. So they're actually circulating that cerebrospinal fluid inside of the ventricle, which is important for keeping the concentration of nutrients and other things the same, consistent inside, or as necessary inside of the ventricles. moving it around, replenishing it, adding things to it, removing things, all of this stuff. These ependymal cells are in charge of that. Okay. And finally, oligodendrocytes. These are dendrocytes. These are going to be your, excuse me, your cells that are actually going to be in charge of myelinating the neurons of your central nervous system. So when I say myelinating, what I mean is that they form what is called a myelin sheath. Myelin being kind of a lipid, a white fatty substance that helps to protect the neuron, as well as acts as kind of a conductive wrap around, or non-conductive cushioning, but also insulating, that's the word, wrap around the neuron. So think about a wire, any wire usually that you have in your house, so charging wire, it's always wrapped in plastic, right? Not just because you don't want to get shocked by the current that's traveling through the metal wires inside, but because the current traveling through the metal wires inside will literally escape anywhere if they're not insulated. And that insulation helps that signal. So that insulation helps that signal to travel quickly and stay this roughly the same strength and not be lost out of the wire as it travels. The myelin sheath of both the central nervous system and the peripheral nervous system, which is formed by different cells. So oligodendrocytes form this myelin sheath on the central nervous system, has this important role. Okay. In the central nervous system, we will learn that the brain has both gray and white matter, as does the spinal cord. So the central nervous system has gray matter and white matter. Gray matter, generally speaking, is where the cell bodies of neurons are located. This is the cell body of an oligodendrocyte. So maybe something like this, the cell body of a neuron proper, which we'll get to in a minute. That's usually the gray matter, because these cells in the central nervous system, the oligodendrocytes, form myelin sheaths on the axons, okay, or bundles of axons, also known as nerve fibers, but not on the cell bodies, okay? So the cell bodies are kind of gathered up in the gray matter of the central nervous system, and then their axons reach down into the white matter of the central nervous system, and we call it the white matter because it's covered in this fatty white lipid myelin sheath formed by oligodendrocytes. So this, all the blue in this picture is the oligodendrocyte. This is the cell body of the oligodendrocyte, and that's its nucleus. So it's reaching out and wrapping multiple axons or nerve fibers from multiple neurons, right? Okay. So that's in the central nervous system. Now let's think about the nerves that are branching off of your spinal cord in your peripheral nervous system, okay? Both the efferent and the afferent pathways, right? So both entering the central nervous system and leaving the central nervous system. You've got different cells performing kind of these kind of similar functions for the cells, for the neurons in the peripheral nervous system. You've got satellite cells, which are cells that are going to cover the bodies of the neurons in the peripheral nervous system. And they're going to perform similar functions as the astrocytes do in the central nervous system. So these have a protective function. They're going to help to maintain the concentration of and the homeostasis of both the intracellular fluid, that's cytoplasm inside of the neurons, as well as the interstitial fluid or the extracellular fluid surrounding all of the neurons, maintaining that. Otherwise maintaining the health and position, the physical position of these neurons. And then you have Schwann cells, which are basically the oligodendrocytes of the peripheral nervous system. They form the myelin sheaths around the axons of neurons in the peripheral nervous system, okay? So satellite cells are basically the astrocytes of the peripheral nervous system, and Schwann cells are basically the oligodendrocytes of the peripheral nervous system, okay? The Schwann cells of the peripheral nervous system also help to regenerate damaged peripheral nerve fibers, which are, generally speaking, more amenable to being repaired after damage than the neurons in your central nervous system. More on that in a bit. All right, let's talk about how this myelin sheath actually forms from the Schwann cells of the peripheral nervous system, right? So in the central nervous system, those oligodendrocytes, they've got one cell body, then they reach out and wrap themselves around multiple axons of multiple neurons. That's not really how Schwann cells work, largely because you don't have like a jumble of neurons going on in your peripheral nervous system. Generally speaking, your peripheral nervous system is all just like nerve fibers, right? It's all going somewhere. It's not, they're not jumbled on top of each other, like sharing information with each other. They're pathways that go from the spinal cord out to the rest of your body and from the rest of your body to the spinal cord. So they're kind of just lines, okay? As such, the Schwann cells that form the myelin sheaths around them only need to cover an axon in its length, right? So, or a nerve fiber bundle in its length, down its length, right? So it doesn't make as much sense to have a cell body reaching out to multiples. Instead, you're going to have a cell that wraps itself around the axon, one axon at a time, or one bundle of axons at a time. So the Schwann cells are actually these in yellow, and each one is an individual Schwann cell, all right? The nucleus is visible inside of it, and the rest of the cell body flattens out and is filled with this lipid substance that is the myelin sheath, is basically the cell body that wraps around multiple times around that axon. So you have multiple Schwann cells wrapping around this axon of this neuron, okay? In between the Schwann cells, this is important, believe it or not, you have what is called the node of Ranvier, which is very French, right? So the nodes of Ranvier are the gaps in between the Schwann cells, which will be very important when we talk about how signals are propagated down the axons of neurons in the peripheral nervous system. So the myelin consists of multiple layers of cell membrane of the Schwann cell, okay? Oops. My pen. Okay, so this is the Schwann cell. This is the nucleus of a Schwann cell. And this in there, inside, is the myelin, okay? So in the peripheral nervous system, you have Schwann cells forming myelin sheaths around long stretches of axons reaching from the central nervous system to the peripheral nervous system, or to the rest of your body, right? Whether it's traveling from sensory organs back to the spine, or from the spine to your muscles to control your body. Either way, we wrap many of those axons in a myelin sheath. And the myelin sheath is actually the membrane, the plasma membrane of a Schwann cell. Okay. All right. Make sure I got this. I'm rewriting these because it's so small. This this picture is from a different text, but the similar picture in your text is on page 397, in case you're wondering. Ranvier. Okay. All right, so if a peripheral neuron is injured, it has the benefit of possibly being repaired in a way that the neurons of your central nervous system a lot of times don't. And this is mostly because the peripheral nervous system is made up of neurons with very, very long axons, because again, they're sending signals usually a very long distance, whereas in the brain, you know, they're kind of talking to each other and it's a very short distance. In the peripheral nervous system, you're going from potentially the spinal cord all the way to the tips of your fingers. So it's a very, very long distance. You have long axons, and there is always a chance that they're going to get damaged, right? There's also less of a chance that your central nervous system will be damaged because it's so protected by your cranial bones, right? And the vertebrae of your spinal column. The nerves of your peripheral nervous system they're vulnerable. They're all like right near the surface and everything, right? So if they were to get damaged, what happens sometimes is that the axon is usually what is cut, what is damaged. And if that's the case, then the end of that neuron away from the cell body is going to disintegrate. So the Schwann cells, as well as the actual axon, that portion of the axon itself, will actually disintegrate, and it will be eaten up by phagocytes and basically recycled. And the rest of it, what's left of it, might actually be able to form new axon terminals at the very end of the axon, right? So new ones of these, but over here. And it may actually be able to be guided back to where it was supposed to be and continue to function. So this is what's so amazing about like repair surgeries that people do. My father-in-law had his lower arm and hand caught in some sort of a metalworking machine one time, and it was literally like hanging from a little bit of flesh. And at the hospital, they were able to set the bone, put the muscle back together, and actually sew each of the major nerves together. And he actually has like pretty much full function of that hand. You can see that they did like a transplant of skin. They did a graft over it because the skin was gone from being burned. And so you can kind of see the cool skin graft on it, but that's really all he has in the way of a scar. And he has pretty much full function of that hand. So it's really incredible what can be done with damaged peripheral nerves, what they can do for themselves and what we can do for them medically, if they get help in enough time. So, all right, let's get to neurons, right? Let's talk about neurons and how they work. So we've talked about all these support cells of the central nervous system and the support cells of the peripheral nervous system. What are they supporting? They're supporting neurons, right? These are the cells of the nervous system. They're the ones that actually do the things that the nervous system is known for doing, which is transmitting signals, right? So your neurons or your nerve cells are going to have some basic components and some basic characteristics that are important. The important characteristics are that they are very long-lived. So once you make a neuron as a developing fetus, that neuron lasts you for your entire life, basically. So once that neuron reaches maturity, they're generally amitotic, which means that they do not reproduce. So if you remember from mitosis, I know you don't want to think about it, but if you think for a second about mitosis, think back and how there was the interphase, right? And there was like G1 and S phase and G2. There is such a thing as G0 phase. And what happens is once that cell reaches maturity, instead of going around again to G1 and S and reproducing its DNA, neurons go into G0, which is basically a holding pattern that some cells go into temporarily, but that neurons go into permanently. And they never leave G0, they never go into G1, they never reach mitosis, so they never actually reproduce. So once you lose a neuron, it's gone for good. They do not reproduce themselves, it will not be replaced, okay? So take care of your brain cells. And finally, they have a high metabolic rate. So neurons require a lot of juice. They require a lot of energy in order to stay alive, obviously to do their work, but even just to stay alive. So neurons cannot survive a lack of oxygen for more than a few minutes, right? And of course, there are always those really cool like miracle cases where like kids have fallen into an icy lake and been without oxygen for 10 minutes and they come back because of the cold. So things like that are possible, but generally speaking, it is your neurons that are at the greatest risk of lack of oxygen. and brain damage can result after just a few minutes being without oxygen. And I know that there are people that can hold their breaths for like inordinate amounts of times, and those folks are different. Those folks are special. Not that you couldn't do it, right? Anybody usually, almost anybody can do it. It's just takes a lot of practice, and it's just a matter of lung capacity. Okay, so the parts of a neuron, generally speaking, they are the same, but they might be arranged a little bit differently. We'll see that in a second. So the cell body of the neuron, of course, is going to be the big, fat, chunky bit with the nucleus inside of it, okay? So the cell body is where the nucleus is. Branching off usually from the cell body are these processes called dendrites. Remember, a process is something that sticks out, right? So the processes on usually the cell body side of a neuron are called dendrites. Cell body, the dendrites. These guys are in charge of receiving information. Okay, so they receive a signal from a previous neuron or from a sensory organ. Okay, so the very tips of the dendrites of a neuron are known as the dendritic spines. On the other side of the cell body, you have one long skinny process, and that is the axon. And this red arrow indicates the direction that the signal travels down a neuron. And this is true of every neuron, okay? The signal will only ever go in one direction. This is extremely important, okay? So a neuron is going to be an afferent neuron, or it's going to be an efferent neuron, and it's not going to be both. There are neurons that send the signal. That's why we call them two different things. The afferent division of the peripheral nervous system consists of neurons that only send the signal from outside to the central nervous system. The signal only goes in one direction. And your efferent neurons are neurons that are arranged that the signal goes from the central nervous system out to the rest of the body, only in that one direction. Okay, so basically the neurons are just oriented oppositely in afferent and the afferent and the efferent division. Okay, the cell bodies are closer to the CNS in the efferent division, and the cell bodies are kind of out in the peripheral nervous system for the afferent division. Okay, but in each individual neuron, the signal only travels in one direction. Okay. Finally, at the end of the axon, that signal gets passed on to the next axon through the synaptic or the axon terminals. Okay, you may see them called either. I will accept both, obviously. Okay, so dendrites are the are the ones that receive the signal from whatever. The cell body is where the nucleus is. The axon is the long skinny tail, and the axon or the synaptic terminals is where that neuron sends the signal off to the next neuron or to the muscle organ or gland. Here's another picture, also showing that directional flow, right? So that signal goes in at the dendrites, it goes through the cell body, it reaches the axon hillock, which is a very important structure. This structure is basically just where the axon meets the cell body. And that also happens to be the place that decides whether or not this signal that has come in so far is worth sending all the way down the axon and to the next neuron or to the muscle or whatever the next structure is. Okay, so what the axon hillock is going to come into play here as we describe this process. So check out this little neuron right here. It received a signal. It decided it was important enough to travel down the axon, sends it on down the axon, and its little axon terminals form a synapse with the cell body or the dendrites of the next cell. This neuron now is going to receive that signal and possibly continue it on, pass it on to the next neuron or muscle or whatever. Okay, We also can see the myelin sheath here, which is nice. Right. Okay. Oh, this also is showing you these terms presynaptic cell and postsynaptic cell. So if the synapse is basically the place where the two cells are talking to each other, where they meet, then one is going to be after or postsynaptic, and one is going to be before the synapse, presynaptic. Okay, so the presynaptic neuron is the neuron that is giving the signal, and the postsynaptic neuron is the neuron that's receiving, or that is receiving the signal. Presynaptic sending, postsynaptic receiving. So this has to do with like each and every cell junction, okay? Each and every synapse. One is going to be pre-synaptic, one is going to be postsynaptic. Okay, so let's talk about the different types of neurons now. There are 3 different types, and then there's also different structural elements. As you can see, we do have some different looking neurons that do not quite fit the profile of what we've usually normally seen as neurons up to this point. But that being said, generally speaking, you have 3 different types of neurons. You have your sensory neurons, right? These are going to be your afferent, part of your afferent division, right? These are the guys that are going to be taking the signal from your sensory organs and sending them to the central nervous system, okay? So your sensory neurons are taking sensory information from your sense organs and sending it to the central nervous system. The interneurons are neurons usually that are within the central nervous system, and they are going to basically be the neurons that are in between the peripheral nervous system and where that information is actually going to be processed, okay? So the interneurons are in the central nervous system. They are going to receive the signal from that sensory neuron, and then usually pass it on to another interneuron, okay? So you basically have, if you're, say you touch a hot stove, right? The sensory organ is gonna be those little temperature receptors in your skin. Those temperature receptors are going to send the signal to your sensory neurons. The sensory neurons will send it to your central nervous system. Usually they'll pass it off to an interneuron, and then it'll be like interneuron, interneuron, interneuron until you get to the brain, okay? And then in the brain is a lot of interneurons. And then once the brain decides what to do with that information, AKA my hand is burning, I'm about to be permanently damaged, it's going to then send a signal from the central nervous system down other interneurons to motor neurons, which send a signal away from the central nervous system to an effector, which is probably going to be, in that case, your biceps and your triceps and other muscles of your arms and hands to pull your hand away from the hot stove, right? Away from the CNS, okay? So these guys are efferent, part of the efferent division, right? Okay, so sensory neurons can have this funny body shape, which seems weird, but we'll talk about it in a second. Your interneurons might look something like this. They're short and squat because they're all bunched in together, right? In like in the gray and white matter, usually the gray matter of your brain. And they talk to each other all very close to each other, right? So they don't need these long axons to reach each other. They're all right there. So they have relatively shorter axons. And your efferent neurons are the ones that need to go from your central nervous system all the way out to your muscles and organs and glands, and therefore have nice long axons, right? The sensory neurons also have nice long axons. They're just arranged kind of weird. So let's talk about these weird structural differences that these types of neurons can have. Okay, so these, your efferent, usually neurons, according to this, right, are kind of the classical looking neuron setup, right? You got the cell body and the dendrites at one end, a long axon, and a bunch of axon terminals at the end of the axon. These guys are what is called multipolar, okay? So multipolar neurons are the neurons, like the classic neuron that you think of when you think of a neuron. These guys right here, which look like sensory neurons, and as is the case, of course, they are usually sensory neurons, are what are called bipolar neurons. Okay, because they've got the cell body right in the middle, and they've got, again, an axon, but over here they have one fused dendrite. One fused dendrite. Okay, so basically the dendrites are fused a long ways until they branch out, which makes them kind of look like an axon, but it's not. Okay, so bipolar neurons, they're usually sensory neurons, and they have an axon and one fused dendrite on the other side. Finally, the really weird ones, you've got your unipolar or pseudo-unipolar neurons, which have one long axon with the cell body kind of like branching off from it. So basically, the cell body is out in the middle of the axon instead of it being in between the axon and the dendrites, which is kind of weird. So the reason why you might have bipolar and pseudo-unipolar neurons is usually in sensory neuron situations. Uni or pseudo-unipolar neurons. These guys are usually bipolar, and these guys are your multipolar. The reason why your sensory neurons are pseudo-unipolar or have this conformation here is because, like I said, the neurons that are traveling from the peripheral nervous system to the central nervous system, where do their cell bodies live, right? It makes sense that in the efferent nervous system, part of the peripheral nervous system, the cell bodies of those neurons would be in the spinal cord and the axons would reach out to the organs, right? Because that's the direction of the flow of the signal from the cell body out along the axon. So if we're traveling in that direction, it makes sense the cell body would be in the central nervous system and the axon reaching out in the efferent nervous system in the case of motor neurons. However, sensory neurons that are sending the information in the opposite direction from sensory structures to the central nervous system, where do their cell bodies live? They live in the middle of their axons, or have long dendrites. Basically, they're going to be along the path and they're going to be passing that signal along to each other. These, where the cell bodies of afferent or sensory neurons are gathered, can be called a ganglion. Okay, so when you hear the word ganglia or ganglion, that's usually where a bunch of these cell bodies are like hanging out because they don't have anywhere else to be while they're like passing the signal from the peripheral nervous system, from sensory organs to the central nervous system. Okay, so again, signal only goes in one direction, right? It's hard to tell for a lot of these bipolar interneurons which one is the axon. So the signal still goes in one direction, but it's usually pretty hard to tell which direction that is. If that is the axon, then the signal is of course going to be going in that direction. Signal will be going in that direction there, if that's the axon, right? And for somebody like this, it's really kind of impossible to know unless you're able to like actually, put some sort of a monitor on that cell, the direction of the signal, or if you know, where the signal came from and where the signal is going. But for efferent and motor neurons, it's pretty clear, usually. Okay. Here again, this image, this is also from your textbook. This is on page 398. This is just showing that, again, you've got your receptive region, the one that takes the signal from the previous cell in blue, the conducting region, where the signal is transported in green, and the secretory region, or where that signal is passed on to the next cell, in pink or red, whatever. Okay, so basically all that signal always goes in the same direction in these cells. Just these zones look a little bit different in whether or not it's going to be a multipolar, bipolar, or unipolar cell. Okay, so again, also this is important. You're going to have your multipolar cells as the most abundant in the body, the major neuron type in the central nervous system. Again, your strange bipolar cells are relatively rare. They're going to be found in some special sensory organs like your eyes. And your unipolar or pseudo-unipolar cells are usually found in the peripheral nervous system. These are the guys that are going to be transporting sensory information to the central nervous system. Okay. And their bodies are forming root ganglia or sensory ganglia. Okay. Okay, which we'll talk about more when we talk about the actual structures of the nervous system. We still have all the nerves to memorize. Woohoo! We won't do that until next week, so don't panic. All right, so again, here's some more variations. So these things don't always look, textbook. There's gonna be a lot of different variations to this. Your multipolar ones, this one's very pretty and everything, but usually they look something more like this. They're in the central nervous system. They're forming these gigantic webs of dendrites, right? Or, gigantic webs of dendrites and an axon somewhere in there. Your bipolar cells can be like kind of weird and chunky, or they can be long and skinny, kind of textbook like this. And your unipolar or pseudo-unipolar cells, I'll accept either term. They mean the same thing as far as this class is concerned. Could be, long or short or whatever. Okay, so just some examples. So some key terminology here that I've already kind of gone over. Peripheral nervous system. So that should be central nervous system versus peripheral nervous system terminology. Bear with me, you guys. I just wrote this PowerPoint. So you guys are the lucky ones. All right. So the nucleus, of course, or in terms of the central nervous system as a whole, is going to be a collection of neuron cell bodies. Okay, so a nucleus for a cell, of course, is the information center where the DNA is kept inside of a single cell. But when we're talking about the central nervous system, you may see the term nucleus referring to a group of cell bodies of neurons all together. Okay, so nucleus generally just means like the center, right? So a nucleus can be a group of neuron cell bodies all gathered together. A collection of neuron cell bodies in the peripheral nervous system is called a ganglion. A bundle of axons in the central nervous system, okay, so like the pathways that we talked about between like the synaptic pathways, right, in your brain, that type of thing, is called a tract. These pathways are called tracts. But a bundle of axons in the peripheral nervous system away from the spine throughout the rest of your body is just called a nerve. Okay, so we don't really call them nerves in your head, we call them tracts. And ganglia are what we refer to where we find the cell bodies in the peripheral nervous system, because they like have to find places to be, right? Got to find places to put these guys because the signal's going in kind of the opposite direction of what you would expect. Okay, you guys, so I'm going to wrap that up for us for our first part of chapter 11. Stay tuned for part 2 whenever you're ready, and I will see you there.